And we know there's some traits associated with that there related to how fast that shell hardens. But if you look at the European studies, they weren't finding a whole lot of correlation between shell thickness and damage at harvest. So some of the things that we've been trying to do is some basic things like identify what type of weevil we have here, learn a little bit about its life cycle. What we'd like to do is eventually develop a model to forecast hazelnut weevil obtusus abundance on the hybrids. Figure out a little bit about, when are they correlated with appearance, given the plant's phenology, the multiple genotypes that Lois has been planting. We've been trying to identify, or working towards identifying resistant genotypes of these hybrid hazels. And borrowing heavily from some of these studies like Guidon's in Europe, to determine if prevalence of these insects is predicated by, traits that we can measure: shell thickness, hardness, volume of nuts, size of hazelnut cluster, easy to harvest, and working through those. So real quickly, here's a few things that we've done this past summer amid the pandemic. We were able to get out weekly from late May into early July and sample for weevils. Working with Lois, we picked out 5 genotypes at each location in Rosemont, worked on the St. Paul campus as well, 2 plants of each genotype. And we would sample from both in-row sides of the plant. And Haley had a video that's not working with the PDF compiler that I'm using on this computer here, but you basically take a big sheet and you stretch it out on a frame and you hold it under the plants, and then you can take a PVC pole and you can bend the plant and kind of beat it slowly, and you do that a standardized number of times, and the insects will fall out of the leaves onto your beat sheet. And then really quickly, you can descend on them like vultures with plastic bags and forceps, and you can catalog what you have. You get a lot of spiders when you do this, but you get a lot of weevils too. And it's a great way to sample adult weevils. At the same time that you're doing this every week, we were measuring all sorts of phenology characteristics of the developing plants and looking at how big these clusters were, the shell thickness, the hardness, cluster weight, and height. And then at the end of the year, we would harvest a subsample of 10 nuts from each plant at each site. And we would take them to these tables and crack them open, and we'd try to figure out how many weevils each of these nuts had. And collected our data and such. And so some of the things that Haley's been doing now through the fall and the early winter is working on comparing the abundance between sites, between genotypes, trying to determine, are there actually differences that we're seeing between genotypes? And she's working her way towards these different characteristics too in the nuts and trying to determine, is this correlated with weevil abundance and mature eggs as they appear in the females? Some results. We found about 1 in 5 nuts actually had weevils. These sites were both consistently infested. St. Paul, Rosemont, we were getting just over 20% abundance of hazelnut weevil larvae in each of the nuts. So a good year for weevils, not necessarily for the nuts. We found that the prevalence was consistent between sites, but the abundance through time too didn't really seem to differ between sites. We started sampling at the end of May, and we were already finding between 1 and 2 weevils in our beet samples. And they were consistently prevalent right through the spring up until about the 4th of July. They would come out fairly early, and then generally after about the holiday weekend, they would tail off. And we feel good about characterizing this side of the distribution when they were there. I have to say, we were really surprised how early we caught them. Looking at some comparative studies in Europe and out west, we were kind of conditioned to see weevils coming out midsummer. Like, we really don't have to really get on top of sampling these until late June. That's when you'll see a lot of the females on the nut clusters. And Haley is really sharp, and she was out sampling for big bud mite that I'm not going to talk about today. And she noticed right before Memorial Day in May that she's like, there are weevils all over these plants. So that's why we started sampling this early, right before beginning of June, and we were catching insects. And it seems that the obtusus that we have here in the Midwest do tend to come out fairly early in the spring. It's very likely that they're doing some maturation feeding for a period of weeks, doing some mating, letting the eggs mature before they start ovipositing in the nuts. And generally, that oviposition is what attracts our attention, but it's pretty clear that they're out a little bit earlier than some of the other species in kind of what we anticipated. So, kind of a lesson learned for next year and something that we're looking at from a management perspective as well. And so, the other thing that Haley has been doing is taking a subsample of these females back, killing them, dissecting them, and trying to determine how long does it take for the eggs to mature and when do they actually start laying eggs in the nuts. So we'll learn a little bit more about their life cycle by this time next year. One thing that was quite clear this year is that there are some pronounced differences between the different genotypes and cultivars at each site. And I'm sure Lois could give some background on, what might be special about, Rosemont 11.8 or CC40 or Dort 75. But there do seem to be some fairly pronounced differences. Some of these genotypes have more weevils than others, and it seems to be almost a little bit bimodal at times. You're either preferred or you're not so preferred. But we saw these differences at both sites, both in Rosemont and St. Paul. Some plants have weevils, others, we know even before we do the beat sheet that they're just not going to have very many weevils. So some work in progress. We're really wanting to develop some phenology models. We're looking forward to this next spring to see how much variation there is between years, see if we can pick up a 1 year versus a 2 year life cycle. We're determined to get out there before Memorial Day this weekend. They surprised us last year, but they're not going to do that this year. We really want to know when those eggs are developing, like I said, figure out when they're ovipositing in the nuts, in the kernels. We're also exploring some of the importance of some non-curculio species. We did find some little tiny green polydrusus weevils that we don't think are very important in the system, but we want to make sure and not just think. There's another species there that's not a hazelnut weevil that is fairly abundant though too that you can find on these plants. And if it's just nibbling on the leaves, we're okay with that, but we want to make sure that that's all it's doing. And eventually, as we get a couple of years of data across these sites and genotypes, we really want to be, working towards this prize of, are there associations between nut characteristics and weevil prevalence? And just kind of a sneak hint, it seems like shell thickness really seems to make a difference. And that would make sense, given that the eggs have to hatch and the larvae has to tunnel into the shell. So if you can get a nut with a thick shell, early on in its development, that does seem to make a little bit of a difference. So I'm going to stop sharing there. We have very many people to thank as we've dived headfirst into the system. Again, really appreciative to be a part of this initiative. Happy to contribute. We're really enjoying learning. This is kind of a fun new system to be involved in, and it looks like a great crop. So I'm going to stop my share. All right. Did anyone have any questions that they'd like me to field? Or we could go back to Lois.
I have a question, but I will let others go first if they do.
Brian, how long might it be till you have a degree day model for the life cycle of these curculio?
2 years, 48 months.
There's a question in the chat.
Gary White. Oh, hi, Gary. Are there any control methods? Not at present. No, no. I mean, when they get really bad, you could do, contact insecticide spray, but I should say no control methods targeted towards Midwestern populations at this time. If you're in Europe, when you've got things like big bud mites, weevils, broadcast insecticides. But what we'd like to do is learn a little bit more about when they're prevalent, what the spray timing might need to be, just basic sampling things too. Haley's done some really nice work cataloging the abundance through time across the field. And so we've done some elementary spatial analyses and trying to determine, are they coming up from underneath the plants like we'd expect every year? Is there also migration pattern coming in from elsewhere, like a Leptoglossus occidentalis, in seed and cone orchards that we see? Yeah, no targeted control methods at this point beyond broadcast insecticides from time to time.
And it sounds like insecticides would be possible sprayed in June in that period before they oviposit. But you still need to do more research to be sure of that. Is that correct?
Yes.
Yeah. So my question is, Haley has been contacting me frequently with requests for data because her work at Rosemount piggybacks on, uses plantings that I have been using for research for other purposes. And she has been asking for data going way back in time, including about nitrogen fertilization. And my first reaction was, how in the world could nitrogen fertilization affect weevil feeding? But Phil Rudder, who of Badger Set Research Corporation, he used to say that weevils were mostly a problem in acidic soils. Well, if you over fertilize nitrogen, which I probably have in some of my plots, because when you're doing research, if you don't over fertilize, you don't find the optimal level. If you don't over fertilize in some of the plots, you don't find the optimal level. And over application of nitrogen results in acidification of the soil. So I'm wondering if that might be something to consider as well. Now, I've never really put much stock in that idea, except we noticed that weevils at Rosemount in the nitrogen trial there were especially bad this year. Now, there's another reason why they could have been bad, and that is that we also culled some of the plants. So it could be that the weevils in the soil from the plants that were culled then became more concentrated on the remaining plants.
Yeah, and there's a number of hypotheses that could explain why there might be more or less weevils in fertilized trials. Could be something in the soil chemistry, although I'd like to believe that so many root feeding insects that disappear below the ground, they're pretty hardy. And these hazelnut weevils, I'd be surprised if it was soil chemistry. I mean, it definitely could be. I'm not a soil chemist, but I tend towards wondering whether it's something in the plant chemistry itself. It's the old dilemma of a plant, from Dan Herms, to grow or to defend. So, is there something that, spring application of nitrogen, is it doing something to the developing nut cluster, perhaps, making it more vegetatively vigorous, lowering the tannin concentrations on the outside of the husk? It's an anti-feedant or something in phenolics, that might deter weevil oviposition, site selection. Not sure.
Right. Well, it could be that something's happening in the soil chemistry that is harmful to any sort of antagonists that the larva might have in the soil.
That could be too. Yeah. Yeah. If there are like entomopathic fungi or something in the soil, yeah, certainly a possibility. Very good. I see that Connie just put in the chat that the annual hybrid Hazelnut conference is coming up March the 4th. Registration is free, and Haley is going to be giving a presentation there. So if you'd like to see even more on hazelnut weevils and some more incredible illustrations, register for the Midwest Hazelnut Conference.
Mhm.
And with that, I'm going to run off to my faculty meeting.
Okay. Thank you very much, Brian.
All right, you're welcome. Thank you.
I'm excited to see how Haley's work takes shape. So, Cody,
Yep. Do you want to reshare, Lois?
so I need to go back to Zoom and hit reshare again.
Yep, share screen.
Okay, I need to find it there. Am I back to the same settings I was at before?
Yes, you are. It looks good. Go ahead.
Okay, well, I think I'm going to, well, I'll finish talking about this one, but then I'll skip through some of the others. So basically, I was breaking it down by full-set families and pointing out the families that had the largest kernel weight. You see the dotted horizontal line is the minimum by Oregon standards. So we've got 3 families that produced higher than the minimums, but this dotted line is the average of our top 8 selections from the paper we wrote a while back. And you see most of them are improved. So whether or not we need to use the Oregon standards as our minimum is an open question, and I would say probably not, given that our markets probably have more tolerance for smaller nuts. But what's interesting, so I was commenting about the fact that we simply have a lot of this cross, so that might explain why this Eric 4-21 times Jefferson cross, that might explain why there are so many of the top progeny that fall in that cross. So the 3 families that have boxes around them here are the 3s with the greatest number of top individual plants. What's interesting is that Eric 4-21 is a parent of 2 of them, and Jefferson is a parent of 2 of them. So we've got a genomics postdoc who's going to start work on our project on March 1st. So I know that he looks for large families to do his genomics analyses to look for segregating genes. And this family will be one that he's working with. But I was hoping he'd be on this call so that he could tell me whether these families, although there aren't many individuals in them, whether they might fit in with the equation, the calculations. Okay, there we go. So the next slides, I'm basically do the same process, but lumping it by paternal parents. So here we've got the best ones is Jefferson, McDonald, and York, all of them European selections. And then here's by maternal parents. And again, we've got 3 that seem really good. So anyway, I keep on doing that with the different parameters. Percent kernel, it really doesn't seem, the results are not as dramatic. They're much more broadly distributed. And as I said earlier, it seems that crossing to Oregon hazelnuts doesn't have as much of an effect on percent kernel.
Jefferson is also thick shelled.
Okay, all right,
Jefferson's like 36% kernel in Oregon.
yeah. So looking at yield, here again, we've got the European crosses. The best plants happen to have paternal parents that are Europeans, but the Midwest are pretty good as well. So as I said earlier, it's a matter of time before we can really make good conclusions about that. I'm going to jump forward to the breeding index, which is kernel yield times weight. So again, this Eric 4-21 times Jefferson cross looks to be the best, but we've got some others. This SBC B2-7 times McDonald had a great individual, but you know what matters is really the best plants, not the best parents, unless we're going to use these as the basis for making more crosses. And before we do that, I'm going to have a lot of guidance from Scott Brainerd, the postdoc over at UW-Madison. So, so in those figures, we saw lots of differences in family sizes. And this table tries to correct for those differences in family sizes. And by doing that, so the top 20 plants appear in 20% of the progeny from this Oregon State parent OSU 1156.105. Whereas Jefferson, it's only 5% of the progeny are in the top 20 for kernel weight. So I did that for all these parameters and also for the breeding index. And that same OSU parent came up as being the top parent or the most frequent parent in all of those. So I'm glad I did this because I would have overlooked that parent if that hadn't been the case. So we might consider using that parent in more crosses. Looking at maternal parents, the top one, we've got 4 different top ones depending on the parameter, but Eric 4-21 is one of them. Stape N7-6 is another one that I personally am excited about, partly because we've got it in some of our agronomic trials and it looks good there. Undesirable qualitative traits, I don't need to go into this in great detail other than to say that the best parents for the quantitative traits did not have problems with undesirable qualitative traits. So that's one thing that we don't have to worry about right now, at least. So our next steps are to continue to collect data on these plants for at least 2 or 3 more years. And at some point, we might use the top selections as pollen parents for new crosses. One of my questions is, how soon should we do that? Should we do it like this year, or should we wait until we have more data? One thing we are already planning to do is put the top 3, if possible, into micropropagation with our colleagues at Night Hollow. It just takes so many years to get new selections established in micropropagation that we figure we'll get started now. And if we later decide that those plants aren't any good, well, too bad, but at least if they are good, then we'll have them when we need them. And we've got a hypothesis that they might be easier to propagate because they are 3/4 European instead of just a half European. And it seems that the American parentage is part of what makes them hard to propagate. So I could go on and talk about the pollinations that we did last year, but that's just more showing you what our plans are, because of course we haven't planted those out. but I think I should stop and take questions. So I do want to show you one slide here. So what happened to those 2012 and 2013 progeny that didn't perform? In some ways, I feel like I plowed under my babies, but heck, you got to make space for new plants.
And we have a grower named Norm Erickson who we're indebted to for the name in the bottom left corner for what to call those plants. It's a technical name.
Thank you.
So Lois, tell us about the stability of these hybrids that are 3/4 European to the blight.
Well, I do have slides about those. If you want, I can go back to
I don't
that.
want to see slides, just give me the bottom line.
Okay, the bottom line is that yes, there's a lot of EFB out there and it's highly variable, but there isn't nearly as much as we feared there might be. We inoculated and we scout for EFB. It's a little tricky scouting for it because it's very easy to overlook the disease. But we did not use known susceptible pollen from Oregon. We did this project wanting good plants, our objective was not to do heritability of EFB resistance studies. And so
they sent you pollen from germplasm that had resistance.
In
yes,
Oregon.
yes. It had resistance in Oregon, which does not necessarily mean it had resistance here because we have more different strains and more diverse strains here. Some of what they sent us has the gasaway gene, which is a single gene, and some of it has quantitative resistance.
How long will it take? How many more years will it take before we know whether we have a problem or not with the blight and these new materials?
The plants in this field have at least some resistance because there's a number of plants that have been entirely killed by it already, neighboring plants. So they've got at least some amount of tolerance, and it could go anywhere from there to full resistance.
All right.
Right. So of the maternal parents, at the time we made the crosses, we didn't knowingly use any maternal parents that were susceptible, but after we made the crosses, at least one of them has developed a pretty bad case of it. We cannot completely rule out that some of them might have a low level of susceptibility. And I did try to parse out, actually, I know some of them have a low level of susceptibility, but none other than that one have a high level. And parsing it out, I've determined that in our progeny, there's no real difference between the parents that have paternal resistance of either, there's no real difference between the quantitative and the qualitative resistance on the paternal side, but there is a difference. The more resistance, the more sources of resistance that we have in the parentage, the better off we are.
So Lois, why don't you introduce Mark, your partner?
Okay. Mark Hammond has been working for me for, let's see, you started in April 2019, so almost 2 years now. And Mark worked for Badger Set for 3 years before he came to work with me. And it has been wonderful getting other perspectives on hazelnuts. Mark is also very much tied in with the hazelnut grower community. So he knows what other people are talking about And that has really made my work a lot more intellectually stimulating.
So Cody, Jerry Cohen had something to say. Set it up so he can ask it directly.
Jay, I think you can just unmute. If you want to. There we go.
You want me to do it live?
Yes.
Okay. There is a lot of confusion about EFP tolerance and resistance. So the resistance genes are mostly identified in the European lines. And I don't know of any evidence that the resistance genes occur in domestic hazelnut, the Minnesota wild type. And that, from all biology I know, looks like a tolerance mechanism, which more than likely is quite different from resistance genes.
Mm-hmm.
So when you talk about when you're 3/4 cross and whether they're EFB tolerant or EFB resistant, you need to be very careful about that, because 1/4 may not give you tolerance. And tolerance is what makes the domestic hazelnut survive in the pressures of EFB.
Mm-hmm.
And so I mean, I think you can bring in the resistance genes from the European lines, and because you're going to have the same problem of EFB that's able to surmount an attack against those resistance genotypes, because that's what they've seen. Tolerance is durable. It's not as impressive in a biological sense, but it does have durability, and it's probably chemically related. You never know. It might be EFB tolerance and insect tolerance might be related. Who knows?
Yeah, Rutgers thinks that they've found our genes from Rush, which is an old American hazel that was involved in a lot of the early crosses that the northern nut growers people did. But I think the point about the American hazel basically being tolerant and a lot of the most of the badger set hybrids being tolerant as opposed to resistant.
Yeah, plant pathologists hate tolerance.
Yeah, but the upshot of it is going to end up being the amount of time it's just going to take time for the plants to really build a track record that they're, for whatever reason, they're capable.
Well, not necessarily. I mean, the Forever Green proposal they ever want to fund for me was to use a metabolomics approach to try to figure out tolerance and EFB tolerance. But
well, in the amount of time that it's going to take to figure out if these plants are worth it based on their yields, if they can maintain those, they're going to have to live with EFB.
But Mark, if you actually had a chemical tracer, if you actually knew the chemical basis for tolerance, you wouldn't have to wait until yield trials are done. You would actually know whether a tolerance mechanism was there. There is an advantage to science-based rather than agronomic-based work. It's not an intractable problem. It's just one that doesn't have any data that people have seriously looked at.
Yeah. So you see, I showed that slide of the greenhouse that was as chock full of seedlings as we could get it to be. And just the past few days, I've been negotiating with the greenhouse managers about greenhouse space, because they wanted to give me a smaller space than I had asked for. And I said, no, I need all that space. So if I could identify which are the susceptible plants and cull them early, that would be a big help.
Like I said, Lois, we've tried to approach that problem scientifically, but but we can only do so much. But I just wanted to say in your discussion, you want to be really clear, because I'm pretty sure that, even though there's not a lot of data there, that the domestic resistance isn't resistance, it's tolerance. There may be resistance genes in that pool. I never say there wasn't, but the thing that you see is more of a durable tolerance, and separated from something where there's actually resistance genes. And there's, ideally you'd have both. Ideally, your ideal cross would be to bring in all the desirable characteristics from domestic hazelnut and the resistance genes from the European. Then you have something then you'd have something that might be insurmountable by the fungus.
Right, right. So this slide that I pulled up, I presume everybody can see Efe Resistance Observation from Rutgers. Tom Molnar crossed 8 badger set hybrids that were, he found them highly durable, highly resistant in New Jersey in a planting which had very, very high levels of EFB inoculum. And he crossed them with EFB susceptible Corylus avellana, and of the progeny, only 19 were disease free after 5 years. That is 96% disease incidence. And so his conclusion was there was something about that cross that caused the EFB resistance. He wasn't talking about tolerance, but resistance to break down. And it was because of that research that he advised me to use EFB resistant pollen from Oregon. So I've also got this other slide showing basically breaking down our data, and you can see how highly variable it is. So we've got the maternal parents grouped at the top and the paternal parents grouped by the gas-away R gene versus the quantitative gene at the bottom. And the pattern is just kind of all over the place. And then the next slide lumps those, and it's still highly confusing. Now, part of the reason why this is confusing is because the information that the folks from Oregon sent me on the type of resistance of each of their plants often said this pollen is from a plant that is an offspring of this plant that has gas-away resistance or that has quantitative resistance, but we don't know about this particular plant itself. So the paternal parent information was rather ambiguous. I could probably write them and ask them for better information. No, they probably didn't have it at the time they sent me the pollen.
Well, Lois, we probably should bring it to a conclusion for today. But before we do, are there any really last burning questions that you would like to put in front of Lois?
We could, this is Les, we could mention that there are a lot more segregating families now in newer plantings at both Rosemount and at Becker. So there's a lot more to look at.
Yeah.
Thanks,
Yeah,
Les. So again, Lois and your team, thank you very much for the update. Looking forward to continued progress with your expanded team. So those that don't know, this partnership between University of Minnesota and University of Wisconsin Research and Extension is a very good collaboration that I think is going to pay off, I hope, in just a few years. So thanks. Jason is also online. So thank you, Jason, for your comments today, and looking forward to this program moving forward over the next number of years. And Dr. Cohen, get your proposal ready for resubmission.
Yeah, I'm very
getting there,
excited
Dawn.
about.
Don't worry.
I'm very excited